Hi guys, welcome to Sourcing Classes. This is Hima Hegde presenting before you the Anthropological News Diary for the week. To begin with, let us look at the topics under light for the week. The week as in 28th of June to 3rd July. So first we have archaeology and the subtopics for the same, followed by the tribal affairs, then the physical or biological anthropology, the same thing continued, and lastly the socio-cultural anthropology. The detailed elaboration of each of these subtopics mentioned will be found in the A&D document, that is the Anthropology News Diary, which is being uploaded on a regular basis in the official Sourcing Classes website, that is www.sourcingclasses.com. Kindly pay a visit. So topics under light for today. Firstly, we have the Dragon Man, then ABO blood group, genome editing in ISS, that is the International Space Station, forest fires and honey output. So firstly, the Dragon Man, also called as Homo longi. So a nearly perfectly preserved ancient human fossil known as the Harbin cranium. The Harbin cranium sits in the Geoscience Museum in the Hebei Geo University. The largest of known Homo skulls, scientists now say that this skull represents a newly discovered human species. A newly discovered human species called as Homo longi or Homo longi or the Dragon Man. The Dragon Man, as you can see in the picture, the reconstructed face, this is a new species discovered. Now the Homo longi lineage may be our closest relatives. They may be our closest relatives and has the potential to reshape our understanding of human evolution. The Harbin fossil is one of the most complete human cranial fossils in the world. This fossil preserved many morphological details. Morphological details as in the shape, structure, size, things like that. Morphological details that are critical for understanding the evolution of Homo genus and the origin of Homo sapiens. The cranium was reportedly discovered in 1930s in the Harbin city in China. The massive skull could hold a brain comparable in size to the modern human beings, but had larger, almost square eye sockets, as you can see, similar square eye sockets, thick brow ridges, very thick brow ridges, almost a very wide mouth compared to the modern human beings, and oversized teeth. While it shows typical archaic human features, the Harbin cranium presents a mosaic combination of the primitive and derived characteristics, setting itself apart from all the other previously named Homo species. Scientists believe the cranium came from a male individual approximately 50 years old living in a forested floodplain environment as a part of small community. Looking farther back in time, the researchers also find that Homo longi is one of our closest hominin relatives, even more closely related to us than the Neanderthals. Even closer than the Neanderthals. This is an important point to be noted. Neanderthals. However, the discovery suggests that the new lineage that they identified, which includes Homo longi, is the actual sister group of Homo sapiens. The reconstruction of human tree of life also suggests that the common ancestor we share with the Neanderthals existed even further back in time. Now the divergence between the Homo sapiens and the Neanderthals may be even deeper in the evolutionary history than generally believed over 1 million years. The researchers say that the findings gathered from Harbin cranium have the potential to rewrite major elements of human evolution. Like one of them is already discussed, that this could be one of the closest relatives to us, even closer than the Neanderthals. So it could easily rewrite the major elements of the human evolution. Their analysis into the life history of Homo longi suggests that they were strong, Robust humans whose potential interactions with the Homo sapiens may have shaped our history in turn. So moving on, we have the ABO blood group. Now the most well-known medically important blood types are in the ABO group. They were discovered in 1900 and 1901 at the University of Vienna by Karl Landsteiner. So here we have the picture of the scientist. So they were discovered by the scientist named Karl Landsteiner. In the process of trying to learn why blood transfusions sometimes cause death and at other times save a patient, in 1930 he received a Nobel Prize. For his discovery of the blood types. Now all human beings and many other primates fall into the category of four principal types, that is A, B, AB and O. These are the blood groups. So this is the ABO blood groups, wherein you have A, B, AB and the O blood group. There are two antigens and two antibodies. Now what are antigens? Antigen is something that is simply a foreign body which will cause our body to create a response, that is to generate antibodies. So antigen is a foreign body that invades our body, in response to which our body will generate the antibodies. So I repeat, there are two antigens and two antibodies that are mostly responsible for the ABO types. The specific combination of these four components determines an individual's type in most cases. Now people with the blood group A type, the A type blood group will have the A antigen and will have the A antigen specifically. So the people with type A blood will have the A antigen on the surface of their red cells. As a result, anti-A bodies will not be produced by them because they would cause the destruction of their own blood. However, if B type blood is injected into their systems, anti-B antibodies will be released in the systems. Anti-B bodies in the plasma will recognize it as an alien and burst the introduced red cells in order to cleanse the blood of the alien protein. Now individuals with type O blood do not produce ABO antigens. They do not produce antigens of any kind of ABO. As a result, their blood normally will not reject or will be rejected when it is given to others with different ABO types. As a result, type O people are universal donors for transfusions, but they can receive only O blood group for themselves. They are called as universal donors, as you all know, but they cannot accept all other types of blood. However, the blood group AB, those who have type AB blood do not make any ABO antibodies. You have to remember that type O do not make ABO antigens, and AB, they do not make any ABO antibodies. Their blood does not discriminate against any ABO type. Consequently, they are universal receivers for transfusions, but their blood will be used when given to people with every other type because they produce both kinds of antigens. It is easy and inexpensive to determine an individual's ABO type, that is to determine an individual's blood group. So how is it done? A serum containing anti-A antibodies is mixed with some of the blood. Another serum with anti-B antibodies is mixed with the remaining sample. Whether or not agglutination occurs in either sample indicates the ABO type. It is a simple process of elimination of the possibilities, more like a trial and error. For instance, if an individual's blood sample is agglutinated by the anti-A antibody but not the anti-B antibody, it means that A antigen is present but not the B antigen. So if the B antigen is not present, it means that the blood type is A. So that's how it works. So moving on, Here we have gene editing in International Space Station. Gene editing in International Space Station. A team of US scientists has developed a CRISPR-based method for studying how eukaryotic cells repair DNA in space. How the eukaryotic cells repair DNA in space. Now eukaryotic cells are the ones in which the nucleus is within the nuclear membrane. They have eukaryotic creatures have well-defined nucleus. DNA in the space. Now such DNA damage may lead to cancer and other determinal health effects. Now they're studying how these cells can repair DNA in space. So because such DNA damage can lead to cancer and other detrimental health effects, raising questions about the safety of long duration space travel. Therefore, which specific DNA repair strategies are employed by the human body in space may be particularly important. The new technique developed by Dr. Sebastian Kraves and his colleagues use CRISPR-Cas9 genome editing technology to create precise damage to DNA strands so that DNA repair mechanisms can be observed in better detail than would be possible with non-specific damage via radiation or other causes. Now using the CRISPR-Cas9 system to study DNA repair in space has several advantages over previously established models. Now first, this system does not utilize radiation or other reagents that cause widespread, non-specific DNA damage and are unsafe to use during space flight. Second, because of the DSB. DSB is nothing but the double strand break. This is the kind of break you do not want. Now this is generated at a precise location. It is generated at a precise location in the genome, and any change in DNA sequence following repair can be easily identified and tracked using methods previously validated on the International Space Station, namely the PCR, that is polymerase chain reaction, and DNA sequencing. It is not just that the scientists successfully deployed novel technologies like CRISPR, genome editing, PCR and nanopore sequencing in an extreme environment, but also that we were able to integrate them into a functionally complete biotechnology workflow applicable to the study of DNA repair and other fundamental cellular processes in microgravity. These developments fill our team with hope in humanity's renewed quest to explore and inhabit the expanse of vast space, or the vast expanse of space. So moving on, we have the forest fires and honey output. Forest fires coupled with soaring temperatures in around the Similipal Biosphere Reserve. The Similipal Biosphere Reserve in Odisha's Mayurbhanj district are likely to affect the honey output, jeopardizing the livelihood of tribal people in the local district. Now, collection and cultivation of raw honey is an important occupation for forest dwellers, especially for around 2000 families belonging to the PVTGs, that is the particularly vulnerable tribal groups like Khadia, Mankadia. It becomes important for them who live near the reserve. Now the state-owned Odisha Forest Department Corporation, that is the OFDC, and Tribal Development Cooperative Corporation of Orissa, that is TDCC, have provided honey boxes to tribal people in different villages near the reserve for honey production. Honey is one of the sources of income for tribal people in the area. An estimated 500 to 700 quintals of honey were produced in the district and supplied across the country under the name Similipal honey. Tribal people, especially the Khadia tribe, will be worst hit due to the forest fires. Now the honey harvesting season is from April to June. Wild honeybees make hives every year on the same trees which tribal people term as the honey trees. Since it is gonna be the same trees every year, they are calling it as the honey trees. They spend more than a week in the forest to collect honey. The honeybees could not have stayed in the jungle when the entire area was engulfed in smoke. So this is the issue. This is one of the issues that the tribals come across with respect to their economical condition, as they're dependent on honey collection for their livelihood. So that's about it for today. Hope you found it informative. Thank you.